For those of you who are wondering, I launched the apparatus and it wouldn't start. So I'm having trouble with Zoom lately. I don't know why. Can somebody confirm you can hear me?
Yeah, we can hear you.
Great. Let me share my screen. Should be seeing my screen now. Before we begin, are there any questions? My computer is going to be a little slow because I'm still running the Zoom session from, I can't show that, from the lecture. We're only 88% done. How do I get out of there? All right, today is October 29th, and we're supposed to be talking about Lab 7. Oh gosh, I'm supposed to send you the unknown number and unknown results today. I'm not quite ready. Hopefully, I can get that out sometime tonight. If not, I'll try and get it out early, like after midnight. So early tomorrow morning, but it'll be part of my night. Any questions about any of that? All right, today we're doing Lab 7, throat and urine cultures. I don't think I need this. I'm going to shut that down. Oop, didn't get it. There we go. Lab 7, throat and urine cultures. Oh, good. The lecture page is finished. Lecture Zoom page, and so my computer will now speed up. All right, Lab 7, throat and urine cultures. There's some reading, as is normal for a lab, not too much. A page talking about the nose, throat, and the mouth and normal microbiota. Um, there's some information about beta hemolytic streptococci, which is actually at the end of this lab module. And there's some information about Haemophilus. We talk about, I think it's Haemophilus influenza in this lab. And there's some text at the end of this lab module as well, discussing Haemophilus. There's also a page to read about the oxidase test, a page to read about the catalase test, and then some information about the coagulase test, which is also at the end of this lab module. Some supplemental material. We have some video clips for you to watch in today's lab. The learning objectives: first, be able to explain how to properly collect throat and urine specimens for routine culture. 2, list common bacterial pathogens isolated from the throat and from urine cultures. 3, know the culture media used to cultivate these pathogens. 4, recognize which tests are used to identify throat and urine pathogens. And then understand the principles of these tests and how to interpret them. We're going to talk about the catalase test, the coagulase test, and the oxidase test, and you should know these tests. We'll briefly mention the XV factor testing. I do not expect you to know that test or be able to explain it in a quiz or something like that. Any question about any of that? All right. Once a specimen is received in a lab, they're logged and processed in a timely fashion. Specific culture media are inoculated and incubated depending on what species they're looking for. We're going to talk about specimens obtained from your throat and urine cultures. Below is an overview of the culture media used to cultivate common bacterial pathogens isolated from the throat and from urine cultures. So first, there's the throat, and we use blood agar as the main agar because the main bacterial pathogen is Streptococcus pyogenes. This is the bacteria that causes strep throat. A secondary agar we use occasionally is chocolate agar, and we use chocolate agar to look for Haemophilus influenzae, especially in small children and in infants. Haemophilus influenzae can infect them and cause problems for them. Haemophilus influenzae, fortunately, is very unlikely to cause problems in an adult or an older child. For urine cultures, we use two media always for the same urine culture. We always use blood agar, and then we use, in addition to blood agar, either McConkey's agar or EMB agar, one or the other. Blood agar will grow everything, including gram negatives, gram positives, and many fastidious organisms. McConkey's agar or EMB will only grow gram negatives. For urine cultures, we always grow it on blood agar and then either McConkey's or EMB because the main bacterial pathogens in a urine culture are gram negatives, especially E. coli is the number one human pathogen in a urine culture, but there's also members of the family Enterobacteriaceae and Pseudomonaceae, which are frequently in urine cultures. But you can get some gram positives in urine cultures, like Streptococcus and Enterococcus and Staphylococcus species, and that's why we use blood agar. Any question about any of that? All right. For a throat culture, sore throats may be caused by either viruses or bacteria. If you see white patches on inflamed tonsils or in the back of the throat, that is indicative of a bacterial infection. The most common bacterial pathogen is Streptococcus pyogenes in a bacterial sore throat. Now, obviously, most sore throats are caused by viruses, and you will not see white patches. And we're not going to talk about a viral infection causing a sore throat. But if you have a bacterial infection, you'll see white patches on the throat, and or usually, and you'll be usually caused by Streptococcus pyogenes. Streptococcus pyogenes does have some other names. I don't use these other names. I use the scientific name, but it's also called Group A Streptococcus, or GAS for GAS, G for group, A for group A, and then S for Streptococcus. Strep throat may be determined by swabbing the back of the throat or the tonsils where there's redness or under white patches, only touching the back of the throat or the tonsils. You do not want to touch the sides of the mouth, the tongue, and it's not listed up here. You don't want to touch the top of the mouth either. And a tongue suppressor can help you do the throat swabbing. Patients that test positive for Streptococcus pyogenes are placed on an antibiotic therapy because a patient cannot easily throw off Streptococcus pyogenes infection. Streptococcus pyogenes has a capsule, and the immune system is not good at throwing it off. Since Streptococcus pyogenes is fastidious, we grow it on a blood agar plate. Blood agar has 5% sheep's blood to facilitate Streptococcus pyogenes to grow, meaning Streptococcus pyogenes will not grow on a general purpose media, such as, what is that called? Nutrient agar. It must grow on something like blood agar because the blood supply what the cells require to grow. You take a look at the blood agar after you've incubated it, and you look for small white colonies with a clear zone of beta hemolysis or complete hemolysis around the colony. And that will be likely to be Streptococcus pyogenes, but it could be another bacteria, especially another Streptococcus species. You only need 2 biochemical tests to make a definitive identification for Streptococcus pyogenes. Once you have beta hemolysis on a blood agar plate, you can run the catalase test and the bacitracin susceptibility test to show that it's Streptococcus pyogenes. So we have a blood agar plate, and this is a blood agar negative, which is showing you mainly gamma hemolysis or no hemolysis. There's a little bit of alpha hemolysis up here, meaning darkening of the media. And then here, let me blow this up so you can see it better. Here we have beta hemolysis, and that small white colony there, surrounded by a very large clear zone, is showing you beta hemolysis, and the white colony is the Streptococcus pyogenes, which I've got to really blow it up. There's the white colony there. And that region there is the complete hemolysis around it. This is beta hemolysis. Once you get that, you run the 2 tests. The first being catalase. Whoops, that's too small. I have to shut this down again. I don't know what I did. Well, it'll work. It's not really ideal though. Um, we'll go to the next slide. Oh, there it is. The catalase test puts the bacteria on hydrogen peroxide, and if the bacteria produce catalase, it'll convert the hydrogen peroxide into water and oxygen. The oxygen will cause bubbling, and if you see bubbles like this, that's a catalase positive test. That's a catalase negative test. Streptococcus, members of the Streptococcus family and the Enterococcus family can grow on a blood agar plate, and Streptococcus could be beta hemolytic, and they will be catalase negative. You then do the bacitracin test. So if it's catalase negative, it's either Streptococcus and Enterococcus. And then you do the bacitracin test. A bacitracin is an antibiotic. You take your swab of the bacteria and put it in this region of the plate, I guess that region of the plate, and then put the bacitracin disc on the plate, and then incubate the plate overnight in the incubator, and then take a look at the zone of inhibition around the bacitracin. If the bacteria are sensitive to bacitracin, then it's a positive test, and this is Streptococcus pyogenes, which is also called group A strep. If the bacteria are negative for the bacitracin test, then it is not Streptococcus pyogenes. And you can be negative showing no zone of inhibition or a zone of inhibition less than a certain size here. which I think I've got in the lab. Another organism that's clinically significant when isolated from the throat of a child, young child, or an infant is Haemophilus influenzae. It's a gram-negative coccobacillus. Haemophilus influenzae is one of the common causes of ear, eye, and or sinus infection in infants and young children. Fortunately, older children are less likely to have a problem with Haemophilus influenzae. And of course, adults do not usually get Haemophilus influenzae infections either. Haemophilus influenzae is a fastidious, tiny gram-negative coccobacillus that cannot grow on blood agar because it needs the V factor and the X factor before it'll grow. And on a blood agar, Haemophilus influenzae cannot lyse the red blood cells. So Haemophilus influenzae will not grow on a blood agar plate. But it can grow on chocolate agar because chocolate agar is the same as blood agar, except we added the blood when the agar first came out of the autoclave, when it is very hot. And that will lyse the red blood cells, releasing the factors, specifically the V factor and the X factor, from the blood cells, which will then make the factors available to the Haemophilus influenzae. So Haemophilus influenzae will grow on chocolate agar as shown here. It will not grow on blood agar unless you do something special, like you put a streak of Staphylococcus aureus this way on a blood agar plate, and then you streak the Haemophilus influenzae perpendicular to the Staphylococcus aureus. And then you can see that Haemophilus influenzae can grow in this region. Let me blow that even further. This region near the Staphylococcus aureus, but it will not grow in this region here. And that's because Haemophilus influenzae needs the factors from the blood, and they cannot lyse the red blood cells. So Haemophilus influenzae cannot get the factors, will not grow in the blood agar, unless Staphylococcus aureus is nearby, because Staphylococcus aureus can lyse the blood cells, which if I blow this way, way up, you might be able to see it. You can barely see it now. There, that central streak is the streak of Staphylococcus aureus, and the white region around it is the complete hemolysis of the red blood cells. By lysing the red blood cells, the Staphylococcus aureus is releasing the factors that are required by Haemophilus influenzae to grow. And that's why Haemophilus influenzae is able to grow in this region of the plate, but will not grow in this region of the plate. Any question about any of that? Normally, we do not grow both Staphylococcus aureus and Haemophilus influenzae on the same plate. We normally will use chocolate agar to grow Haemophilus influenzae. All right, that's it for the cultures that you normally get from a throat culture. For urine cultures, there's more bacteria that can cause a urinary tract infection. Urine cultures are used to diagnose a urinary tract infection. Bacteria can travel from the urethra up to the bladder, and even further. They can travel up to the ureters, and further they can travel up to the kidneys. But largely we'll be talking about urinary tract infection, which is also called cystitis, where the bacteria have invaded the bladder. Females are more predisposed to developing a urinary tract infection than males, and that's simply because they have a shorter urethra. The lower region of the urethra is colonized by bacteria, but the upper regions of the urethra and the bladder are colonized with much less bacteria. I will state that in females, they actually do have a normal microbiota of bacteria growing in the bladder. This bacteria does not normally cause disease for the female. For males, I've never been able to ascertain if they have a normal microbiota in the bladder. They may not because they have a longer urethra and it's more difficult for the bacteria to get up the urethra and into the bladder. However, there's not been any studies done on male bladders that I could find. So I don't know. The question is, do males have a normal microbiota in their bladder? Females do. It's a few species, but these species do not cause urinary tract infections and they do not cause problem for the females. It's part of their normal microbiota. And if they get other bacteria in their bladder, it can cause a urinary tract infection. To assay for urine culture, the genital area must be clean prior to collection. And then you do a clean catch midstream, which involves collecting only the middle portion of the urine stream. If you collect only the middle portion, you get less contamination from the urethra than if you collect urine from the start of the urine stream or at the end of the urine stream. And patients prefer the clean catch midstream to get a urine sample. But you could put a catheter up into the bladder and sample the bladder. The trouble is the catheter has to go through the urethra and it can pick up cells in the urethra and then take those cells into the bladder. So the catheter is not perfect. It's cleaner, but it isn't perfect. And about the most perfect way of sampling the bladder is actually a suprapubic aspiration, where you put a needle through the abdominal wall directly into the bladder and then sample the bladder that way. You'd be very unlikely to pick up bacteria from like the skin because you clean it. You don't really sterilize it, but you clean it before you put the needle through. But patients don't like getting a suprapubic aspiration. And it's because they're getting a needle stuck through their abdominal wall going through their bladder. And they don't really like catheters either. But those are the three ways to get a urine culture: clean catch midstream, a catheter, and then suprapubic aspiration. You then take 1 microliter, that's 0.001 milliliters of urine, and you put it on a blood agar plate using a special inoculation loop. We have a special inoculation loop shown here, which is obviously a very small loop because there's a penny, and that will measure 1 microliter. And then you streak it on the blood agar that way and then across this way. And you look at the growth growing on the blood agar, and then you take a sample and then also grow it on either McConkey's or EMB plate to ascertain for gram negatives. The number one Bacteria growing on in a urine culture is, of course, a coliform, such as E. coli. It's called a coliform. This is a bacteria that tends to be in the colon, such as E. coli, and they're the most common isolates. But you can get other bacteria in the urine culture, and that's why we also grow it on blood agar. The interpretation of a positive urine culture is multifaceted. There's a couple of factors that need to be considered. The type of collection. Was it clean cut voided method versus a catheterized specimen? Because there will be different contamination levels. The gender of the patient. Usually males have less contamination than females from the urethra. And then the number of colonies per mil of the urine. The presence of polymicrobial growth, meaning multiple organisms isolated, may indicate a poor collection. Males generally have about 50,000 colonies per mil of urine, and females greater than 100,000 colonies per mil with voided urine, meaning a clean catch method. But you normally get lower counts from a catheterized specimen. And usually with the suprapubic aspiration, all of the bacteria you get come from the urine. Usually. Once you get the culture of interest, obviously you have to grow it up and get a pure culture. So streak it out, get it up to get a pure culture. You then run some biochemical tests to try and identify it. One test you'd run would be the coagulase test. The coagulase test is looking for the production of the enzyme coagulase. This enzyme is used to differentiate Staphylococcus aureus from other members of the Staphylococcus family. Staphylococcus aureus has coagulase, the other members do not. Coagulase production actually contributes to the pathogenicity of Staphylococcus aureus because it causes the blood plasma to cause a blood clot. And this can protect the bacteria when they're low in number from phagocytosis by the immune system cells, meaning a white blood cell, will have a harder time eating a bacteria in a clot than it would a bacteria outside of a clot. So coagulase can convert fibrinogen into a fibrin clot. And you just look to see if clotting forms. And if it does, then it's the coagulase positive. There's two coagulase tests, a slide test, and if you have a coagulase positive, you'll see clumping on the slide. If it's coagulase negative, there will be no clumping. So it'll just be a smear of bacteria. And then there's a tube test. If there's coagulase positive cells, they will form a semi-solid clot in the plasmin. And if it's coagulase negative, you'll get liquid plasma. And as you tilt the tube, it'll just run. The most common bacteria isolated from a urine culture is E. coli of the family Enterobacteriaceae. There will be other members of the family Enterobacteriaceae, which can also cause a urinary tract infection. And you can get Pseudomonas aeruginosa from the Pseudomonaceae family, which can also cause a urinary tract infection. The Pseudomonaceae family is another gram-negative bacillus causing UTIs. However, unlike the family Enterobacteriaceae, the family Pseudomonaceae, all of the members are aerobic and they do not ferment glucose, and they're all oxidase positive. The family Enterobacteriaceae is a family which can all ferment glucose. They are facultative anaerobes, so they're not aerobic, and they are all cytochrome oxidase negative. So the Pseudomonaceae are cytochrome oxidase positive, and the Enterobacteriaceae are cytochrome oxidase negative. And the oxidase test is the number one test for distinguishing between the members of the family Enterobacteriaceae and the members of the family Pseudomonaceae, although they do have some other differences as well. All right, the oxidase test can determine if it's the member of the Pseudomonaceae family, like Pseudomonas aeruginosa, a human pathogen, from a member of the Enterobacteriaceae family. And the oxidase test will be positive for Pseudomonaceae, showing a blue or purplish spot from oxidase positive. And then an oxidase negative test will be no purple or blue. Like this region doesn't show any purple or blue. So if there's cells there, these cells are oxidase negative. All right, any question about any of this lab? You're not physically going to be performing any of these activities. However, you will be held responsible for knowing how to perform these tests. We have a video talking about the catalase test, another talking about the coagulase test, and a third talking about the oxidase test. So look at these videos and then work on the lab module. Remember, always use the worksheet because sometimes the questions change between the worksheet and the lab module, and I only update the worksheet. As well as the worksheet's the one that should be submitted because it's smaller and it will not fill up my Canvas web page. If everybody were to submit the lab module, my web page could fill up and then no one would be able to submit a further assignment. All right, let me go to the worksheet and then I'll talk about the exercises for Lab 7. So answer the questions. Questions 1 through 3 are looking at the enzyme catalase, coagulase, and oxidase. So in 1A, discuss which enzyme converts fibrinogen into fibrin. And then 1B, name a microbe that tests positive for this enzyme. In 2A, name the enzyme that detects the presence of cytochrome C proteins in the electron transport system. And then 2B, name 2 microbes that test positive for the enzyme. And if you use AI to do this and get the wrong answer, I'm going to mark it wrong, even if it might be correct, meaning you should be using an enzyme from this lab. And we're only talking about the enzymes catalase, coagulase, and oxidase. All right, so 3A, name the enzyme that hydrolyzes hydrogen peroxide into the non-toxic products water and oxygen, and then name 2 microbes that test negative for this enzyme. You can either name 2 species or here 2 families. For slide 4, it asks which rapid test is used to differentiate between the following: Staphylococcus and Streptococcus. And once again, we're looking at these 3 enzymes. And then 4B, which enzyme is used to distinguish between Staphylococcus aureus and other members of the Staphylococcus species. 4C, which enzyme is used to differentiate between the family Enterobacteriaceae, and the family Pseudomonaceae. For 5, concerning specimen collection for routine throat culture, what areas of the throat should be swabbed? And 5B, what areas of the mouth should be avoided? For question 6, name the most common cause of a bacterial throat infection. It must be a bacteria and the most common cause. 7, what test is used by microbiologists to differ between beta hemolytic group A streptococcus and beta hemolytic streptococcus species isolated in a throat culture? So just one test here because we've already narrowed it down to beta hemolytic. And the family streptococcus. And that's why you only need one test, because the other test will only differentiate between the streptococci and the enterococci and other gram-positive cocci. On 8A, answer the following concerning a child's throat culture. State two non-selective agar plates that are inoculated to grow a child's throat culture. One here and then one there. And then 8B, what organism can be isolated from CAP, but not from BAP? CAP being chocolate agar plate and BAP being blood agar plate. For 8C, I've crossed it out because you can't answer it directly from the lab module. You'd have to go online. And you can do it if you want, but you won't get extra credit. All right. Question 9, list 2 media that are used to grow urine cultures. Question 10, list 3 reasons why a urine specimen should be rejected for culture. You can find those reasons in the lab module, although I haven't talked about that. 11, how much urine is placed on the surface of a blood agar plate from a urine specimen? 12, what is the most common bacteria isolated from patients with a bladder infection? And you must name the species here. 13, identify the Gram reaction, what the Gram stain is, and the cellular morphology, the shape of the cell of the following bacteria. 13A, Staphylococcus cephrophyticus. 13B, E. coli. 13C, Pseudomonas aeruginosa. 13D, Streptococcus pyogenes. And 13E, Haemophilus influenzae. Any question about any of this? If not, oops, I lost it now. If there's no questions, go ahead and work on the lab. I will be here for at least until 8 o'clock.